that I want you to appreciate from today's, lecture or lecture is, how do antibiotics work? And we'll talk about general mechanisms of antibiotics. And we'll also talk about how they're becoming resistant, right? And I like that video because it's a pretty good summary and touches on a couple of really important issues that touch on how our use of antibiotics has driven, all right, antibiotic resistance. And even in that horizontal gene transfer that you guys read a couple units ago now, you guys saw, right, how the selective pressure that we're putting on bacteria is causing resistance. And so one of the things that is of concern for us, right, are these superbugs. And this is actually an article that I pulled up to show you guys that is a few years old now, but it's still really relevant. And so this is about a woman from Nevada who had been in India. When she was in India, I believe she fell and broke her hip and had to have surgery, and had multiple or was treated multiple times for bacterial infections while she was in the hospital in India. She comes back to the United States and becomes ill, goes to get treatment, and eventually passes away. And doctors knew what the bacteria was that was causing illness. And you can see here, if you're looking at the article, the bacteria that she had was a type of Klebsiella pneumoniae, which we actually work with in lab, if we were to be in lab. And the bacterial infection that she had was resistant to all 26 antibiotics that were available in the United States. They even tried, let's see down here. I can't remember the name of it. Here we go. Carbapenems, which is like right now the antibiotic of last resort. And there's a couple different variations of them. Once the bacteria that or the bacterial infection that she had was resistant to this antibiotic, they kind of knew that it was the end. So she eventually had bacterial sepsis and organ failure and passed away, right? And so you can see as of 2017, we haven't had a lot of these cases yet in the United States. I haven't checked, again, the state is a few years old, somewhere maybe in the couple hundred. And so not killing large numbers of people, but the fact that these bacteria are out there and are resistant to every type of antibiotic that we have available is definitely concerning. All right. And I also wanted to point out that this pathogen in this patient was identified in something called the Morbidity Mortality Weekly Report, which we'll actually talk about more probably starting tomorrow. But I wanted to show you what that looks like. So here. So these are really cool reports, and I actually have one that I'm going to look through tonight. There's a lot of these out there for COVID-19, but Morbidity and Mortality Weekly Reports basically are where physicians publish articles on interesting cases that they've seen or cases that they've seen for something called a notifiable disease. And so in this case, this is the doctor that was treating the patient, Li Chen. Well, actually, no, Li Chen wasn't the doctor. So the doctor would have been Alexander Allen. But Li Chen was the researcher that was trying to identify antibiotics that would be effective on this bacteria that this patient had. And so they published this report. And these reports are really important for physicians and healthcare workers because they give the nation and the world kind of an idea of what we're dealing with as an illness, right? And so this is basically a case report for this patient talking about their attempt to find an antibiotic that would work. And we'll actually talk about next unit. These reports helped us identify the start of the HIV epidemic within the United States. All right. And so if you go to the CDC's website, you can pull up these reports. They're free. You can download them, and really important for fighting various illnesses and disease. All right. Okay. So let's jump into the chapter here. All right. So antimicrobial medications, and then this will lead or segue very nicely into antibiotic resistance. So if you saw on the video or were paying attention, right, penicillin is kind of the first antibiotic that we commercialize for use. Alexander Fleming discovered penicillin in 1928, and it was because I think he went on holiday, right, vacation for folks that are from the other side of the pond, right? And he left some Petri dishes out. I believe on the Petri dish, he was growing Staph aureus. He came back. There was mold on his plates, and there was a kind of a clearing halo, right, between the edge of the fungi and the bacteria. And so he noted very quickly that the fungi was killing the bacteria. He identified penicillin, but he was actually not successful in isolating or being able to isolate penicillin in large enough quantities that it could be used. And so Cheyne and Flory are two other scientists that come along a little bit later. And in 1941, they figure out how to isolate this in large enough quantities to start treating patients. You saw from the video, the first patient that they attempted to treat was a policeman that got cut in his rose garden, got a bacterial infection, and they started treating him with penicillin. He had immediate improvement. Unfortunately, they did not have enough penicillin, and they mentioned in the video, penicillin is secreted by the body, and so they were collecting his urine, trying to pull out as much penicillin as they could to treat him, but eventually he succumbed. But it was less than a year later when they perfected the isolation process of penicillin from fungi, and they were able to start treating patients, right? And so antibiotics on wide-scale use, right, we're talking about the early 1940s. As was also mentioned in the video that you guys just watched, we see this this co-evolution, or microorganisms, in this case we're talking about specifically bacteria, are evolving as we, right, use new medications. And so that's required to continue continuing to modify or change these drugs. Did anyone catch in the video why he mentioned that there are not a lot of antibiotics in development, even though this is a huge problem?
It's too expensive.
So can you say that again? I couldn't hear.
It's too expensive.
Too expensive, right? And you know, this is one of the things you know, that maybe we don't even appreciate enough right now during this vaccine race for COVID-19, is that it's a huge monetary investment when it comes to drug development, right? And it will be billions and billions of dollars invested from that time when you start researching to the time when maybe you have a product that you can implement or use in public. And if you're talking about a pure profit-driven industry, which I would say the drug industry would fall into that category, if you're looking at an investment, and if I told you, Sarah, I'm going to spend $1 billion researching something so we can distribute it for public use, and bacteria is going to be resistant to the new drug that you make in a year, then you would probably say, well, then why would I waste my time? All right. And unfortunately, that is one of the negative aspects of drug development and pharmaceutical companies, that they are businesses, they are looking for a profit, and antibiotics are not necessarily the most profitable thing for them to be researching. And so one of the videos that I have on Moodle for you guys to watch as he actually talks about in this video alternatives or other ideas to treat or encourage the development of new antibiotics. All right. So we see really early on, scientists begin altering the structure of antibiotics that we already have. And so in the video, right, he kind of mentioned that most of our antibiotics, right, are ones that we've derived, right, or isolated from nature, right? So either antibiotics that we've isolated from other bacteria or antibiotics that we've isolated from fungi. And what we've been able to do is alter the antibiotics that we've isolated to give them new properties. And an example of this would be penicillin, right? And it primarily kills gram-positive. And we actually have altered its structure slightly, and that's where we get the drug ampicillin. And ampicillin is actually a broad-spectrum antibiotic that kills both gram-negative and gram-positive. And so if you look here, this is the penicillin family tree. Right? So you have penicillin down here, and you can see all of these derivatives that have come from it. One of the ones that you guys have probably been treated with is amoxicillin. Is pretty common if any of you have had a Z-Pak. All right. When you go to the doctor, amoxicillin is the antibiotic that's in the Z-Pak. All right. So maybe some of you have had methicillin. So you can see these are all derivatives from, right, this one family or this one class of antibiotics. Okay. So this is just a really simple question, but when we talk about antibiotics, right, if I said I was going to I'll give Rachel D an antibiotic. Rachel D, what do you want that antibiotic to do, and what do you want it to not do?
I want to target the bad cells and not target my cells.
Perfect, right? You want the antibiotic, I love it, right? You want the antibiotic to kill the pathogen, to kill the bacteria, while hopefully not damaging you, right, and your cells. And so, you know, this is something that researchers and doctors are concerned with, right? So what Rachel just said, right? Greater harm to the microbes than to the host. And so when it comes to medications, this is where we introduce the idea of therapeutic index. All right, so you can look up the therapeutic index of any antibiotic. And when we think about the therapeutic index, what we're hopefully trying to do or target is look for the lowest toxic dose possible, all right, that still has an effect on, that still can kill the host, or not kill the host, still can kill the bacteria, right, without affecting the host. So, you know, this idea of, you know, yes, and I actually liked it. Anna was kind of talking about in her article, right, this is different, but you're talking about vaccine doses, and they had that third dose of the vaccine that probably induced an immune response, right, made the antibodies that were needed, but it was having negative effects on the patient, right? And so in the paper that Anna presented, you know, they had those lower doses, and they were able to show, hey, the lower doses, right, create a similar amount of antibodies that a natural infection does. And so let's give the patient the lower dose, right? Because that lower dose gets the effect that we want and is less, has less of an effect on the patient or the host. And the same idea for antibiotics. So we want that lowest toxic dose, right, that still has an effect, right? So a high therapeutic index indicates low toxicity while still being able to kill the pathogen, right? So an antibiotic with a high therapeutic index, this is good for you. This is going to be an antibiotic that you could probably take orally, all right, if that's what was needed. You also, though, have low therapeutic index. And a low therapeutic index does not mean that the medication doesn't work. It just means that there's kind of a trade-off there. It's super good at killing the bacteria, but it's also super toxic or damaging to the host. And for the most part, antibiotics with low therapeutic index are going to be more for a topical application. And so these are not necessarily antibiotics that you want in your system. Some of them can be used in you, right, be given by something like an IV, but these are closely monitored, right? So, and a low therapeutic index antibiotic that is in your system, right, in your body, not just on top of your skin. These are going to be antibiotics that are given in a hospital setting where you can be monitored, right? A doctor is not going to send you home with a low therapeutic index antibiotic for you to take on your own. It requires close monitoring because they are so toxic. All right. When we talk about effective antibiotics, we have to look at two kind of categories, right? So again, you know I love my branches. And so here we have to look at the ability of certain antibiotics to kill versus inhibit growth. And so when we're talking about inhibiting growth, we have bacteriostatic, right? So think about things that are static, they're not moving. So these inhibit growth, but they are not killing the pathogen. All right. And so the idea here would be I give you a bacteriostatic antibiotic. It stops growth of bacteria and it allows, right, your immune system, right, to catch up and clear the pathogen. All right. So you're basically allowing your immune system to do the job. If you think about it, you know, that last video, for whatever reason, like to show the antibiotic or the antibiotic resistant bacteria with like huge biceps, right? And so the idea here would be bacteriostatic drugs essentially are tying the bacteria's arms behind their back, and then you're going in and finishing them off, right? So it's making them an easier target for the immune system to deal with, right? You also have bactericidal, right? And so these, right, are antibiotics that are actually killing the pathogen. So in this case, you're doing what the immune system can't do on its own, right? Your immune system is still responding, but in this case, again, because of exponential growth in that log phase that we've talked about bacteria, maybe your immune system can't keep up, right? There's just too much bacteria. So in this case, when these bactericidal drugs start working, they kill the bacteria and your immune system is still working. So it's your immune system plus this drug, right, that's allowing you to clear these pathogens. Now, one of the things, right, that we need to be careful of, and some of you, actually, we'll talk about this in just a second. We'll get back to this in a second, bacteriostatic versus bactericidal, is that you have to be really careful about prescribing these together. Why would you have to be careful about prescribing these drugs together? Why might it not be a good idea to give a patient a bacteriostatic medication and also bactericidal medication? What have we learned about the basic mechanism of maybe something like penicillin?
If like the cell is like not growing, then the other drug wouldn't know what to attack.
Right. So the basic idea of, you know, if we're talking about something like penicillin that interrupts the development or the construction of a cell wall, and that means that cell needs to be dividing for the drug to work. And so if you were to give a bacteriostatic medication that essentially halts cell division, and you give an antibiotic that actually works well when the cell's dividing, well, then that's not necessarily a good combination. We'll talk about this in a couple slides, but more drugs or just putting drugs together is not necessarily beneficial. It can be beneficial, and I'll point out a specific example of that in a little bit. But just because you're like, hey, these both do good things, giving them together might not give you the effect that you think. We also have to mention, and I'm sure you guys have heard of this before, but it's the idea of broad spectrum. All right. And so broad spectrum antibiotics are going to target a wide range of bacteria, gram-negative, gram-positive, maybe even mycoplasmas, right? They're important to treat acute life-threatening diseases. And so if, let's see, who can I see here? We'll say if Elena, came in and she was obviously very sick, they took her blood and they could tell from her blood based on her white blood cell count that she probably had a bacterial infection. And, one thing that we can do and that that scientists do, right, or doctors do, is they send away a sample, right, of tissue or some type of fluid, or right, or a swab or something, right, from Elena when she came in, and they culture that bacteria to find out what bacteria it is and what medication is great at treating it. However, right, that's going to take probably at least 48 hours to figure that out. All right. And Elena may not have 48 hours. So what do? Well, they give her a broad spectrum antibiotic, and they say, this is going to work until we can figure out exactly what we need to give you. Right. And so this is usually, what we give when someone has a bacterial infection and we haven't identified the bacteria. It just kind of covers all of our bases. One of the big problems here is because it's so great at killing pathogenic bacteria, it's also really good at disrupting your normal microbiota. All right. Now, narrow spectrum, which we're about to talk about, they can disrupt your microbiota also, but not quite to the extent of a broad spectrum antibiotic. All right. So typically, what we use to treat while we're figuring out exactly what bacteria a patient has. Right. Narrow spectrum, limited range, right? And limited range does not mean that they're not effective. It just means that you have to have the pathogen identified. All right. We have certain antibiotics that work great for gram positive, certain antibiotics that work great for gram negative. Even within those different classes of antibiotics, we have bacteria that work better on a certain genus of bacteria. So narrow spectrum, you need the pathogen identified. In the case of, the lady, so Klebsiella, so you have a Klebsiella infection. That's a gram negative bacteria. If a doctor prescribes a gram positive antibiotic, well, it's not going to do anything for that patient, right? So narrow spectrum, you need to know exactly what you're dealing with so that you can treat it properly. So this is something to consider, right? And I mentioned, so this probably should have been the last slide. I had these mixed, but when you're mixing medications, you need to be aware, right, of different effects. And so you have antagonistic, right, additive, and synergistic. So what's antagonistic mean? Amanda, what's antagonistic?
Is it like when you're going after something specifically, like I'm kind of thinking of like a bully that's like actively seeking someone out to like beat them up?
All right, so yeah, so you're thinking about antagonistic from that perspective, right? So these are things that are kind of fighting against one another, right? Bullies and nerds, right? They're antagonistic in a classic sense. Bullying's bad, so please don't be an antagonist, right? But they're kind of fighting each other. They're in opposition. This is not something good when it comes to medications, right? So when prescribing multiple antibiotics, prescribing something that's bacteriostatic versus bactericidal, right? And this is where, if any of you are interested in ever becoming a pharmacist, right? A good pharmacist hopefully catch doctors' mistakes sometimes if they prescribe two things that are in opposition. So in this case, right, these drugs oppose each other. They don't do anything for the bacteria, and the infection persists, right? When we talk about additive and synergistic, this is where I wish I could, draw on a board. But if you think about additive, the way to kind of think about this is more of a linear kind of growth, all right? Synergistic would be more exponential growth. And so, if you have two medications, and I always like to say on a scale of 1 to 5, right? 5 being the strongest, 1 being the weakest, right? You give two medications that fall on that scale, their killing power is both 2. So if we're talking about additive, when you combine them, Caroline, what's their added killing power? If medication A is 2, medication B is 2, what's their additive killing power?
Would it be 4?
It would be 4, right? 2 plus 2 equals 4. Thank you. I like it. Synergistic, all right? If I had a killing power of 2 and a killing power of 2, right? Megan, you're good at math. So synergistic, if I had a killing power of 2 and a killing power of 2, what would my combined killing power be?
Um,
I don't have a specific number in mind. You can just make something that's representative.
Something greater than 4.
Something greater than 4. All right. Way to safely, if we were at the edge of a pool, you just dipped your toe in, but you went in. So I'll take it. Right. 4. Something greater than 4. So, example, I always like to write this on the board. I do 2 + 2 = 27. Right. And so somehow when those drugs are together, all right, it just is able to effectively kill that bacteria in a way that they couldn't individually, right? And so additive and synergistic, when you're combining medications, right, that's what you're looking for. You're looking for a sum, right, that is greater than the individual parts. And sometimes, right, that's just a simply 2 + 2 = 4. But sometimes when you're talking about synergistic effects, that combination goes even beyond the individual values of killing power. And again, that's not a scientific necessarily approach to it, but I think it illustrates the picture of additive and synergistic effects of medications. All right. Now, we've talked about multiple times now, right? You guys pointed out at the beginning of this class, and we talked about it when we were comparing prokaryotic cells and eukaryotic cells, right? Looking for that perfect target. And there is no such thing as that perfect target. There's going to be some type of adverse effect in patients. Hopefully, though, that's minimal. What we do see, though, is that some patients have very strong reactions to antibiotics, right? So allergic reactions, right, are not uncommon. These can be as small as a rash, right, to anaphylactic shock, right, which can lead to death if not treated properly. And it's not uncommon for children to have some type of allergy to certain antibiotics. We found out in the fall, so this last fall, this last academic year, my daughter had a pretty bad bacterial infection, and we gave her amoxicillin, and then she broke out in hives, right? And so we gave her some Benadryl, called the doctor, and got a new antibiotic. And so not uncommon, and those of you that have this, it's in your chart. This is also why, as you get older, if a doctor asks you if you have allergies to medications, you should always speak up in case it's not in your chart or in case they overlook it, right? Toxic effect, prolonged exposure to certain antibiotics can lead to organ failure, inhibited production of red blood cells and white blood cells, which is bad, right? This is going to be low therapeutic index typically, or even prolonged use of high therapeutic index medications could lead to these issues. Is someone doing Lyme disease for their project? Who is it? Does anyone remember who's doing Lyme disease? Speak up.
Rachel and I.
Yeah, there we go. All right, cool. So it's Rachel S., right? Is that who's working with Megan? So Megan and Rachel S., one of the things they'll talk about probably, and if they weren't, they will now, right? Can you see me winking via Zoom? Can you see that, Megan? Okay, good. Is Lyme disease, there's a couple different schools of thought about how much antibiotics and how long these patients are on antibiotics, all right? And so even if the antibiotic is good at what it does, right, kills the bacteria, doesn't harm the host, for a prolonged time, right, or prolonged exposure to antibiotic can even have a negative effect. And then we've talked about microbiota dysbiosis, right? I put up my favorite thing here. This is where C. diff comes into play, and even dysbiosis of the microbiota, though. Professor Perry mentioned in her fungi talk, and I know I have a group that's doing, I think, some stuff on yeast infections, right? Especially in women, right? It's not uncommon at all, right, for a woman to get treated for some type of bacterial infection, right? The antibiotic does a great job of clearing that up. but it disrupts her normal microbiota, and then she has a yeast infection, right? And again, this is not uncommon in women at all, and this would be one of those things, where you wouldn't probably even have to go back into the doctor. You could just call them and be like, hey, and they would just get you some medication to help with that, right? And so there can be extreme cases, right? C. diff is pretty extreme. Yeast infection, although not fun to have, is typically not life-threatening for most folks. All right, so let's take a short break here. It's a little after 9. Let's come back at 9:10. We'll start with basic mechanisms of action of antimicrobial medications, and then we'll talk about some specific medications that are commonly prescribed here in the US. And we'll talk about antibiotic resistance, which you guys already know a good bit about just from the discussion of horizontal gene transfer. So I'm going to go ahead and pause the recording. All right, let's keep going. I think most people are back, and if they're not, hopefully they'll check in soon. Just a little side note, I realized as I was out there drinking my green tea with honey that I misspoke. Amoxicillin is not in a Z-Pak, it's azithromycin, all right? And we'll actually talk about the actions of azithromycin in just a second, but azithromycin is a broad spectrum. And so when you go in complaining about certain things, a doctor, my doctor describes it as the lazy doctor approach. He says that jokingly because his kind of philosophy sometimes is, let's try this and see how you feel. If you don't respond within a couple of days, then come back in and we'll do more kind of stringent testing. So. Cool. So let's talk about what antibiotics are doing. And you guys already know these top two and have even kind of mentioned in your answers from the first exam, whether you know it or not, these bottom three, right? So antibiotics, right? One of their targets will be cell wall synthesis. Now also note too, typically antibiotics are going to focus on one of these targets. So if you want to go after more than one of these 5, you're probably going to have to take multiple antibiotics. All right. So, I don't want us to fall in the trap of like, oh, penicillin does 1 through 5. No, penicillin happens to inhibit cell wall synthesis. And so it's very dependent on, what mechanism or what target you want. All right. So actually here, hold on one second. I'm going to slide so I want to see how much battery I have on my computer. All right. 64%. Okay, good. We're looking good. We're looking good. All right, so inhibit cell wall synthesis. You guys know about that. You guys, way back when on your first exam, well, I usually say way back when during regular semester, that is a while ago. This is only a few weeks ago. But you guys talked about inhibiting protein synthesis, right, by interacting with ribosomes as a good target, right? Inhibit nucleic acid synthesis. And some of you kind of hinted at this on that first exam because you said, hey, prokaryotic cells have a nucleus, eukaryotic cells have a nucleus, prokaryotic cells have a nucleoid, right? They don't have that membrane around their DNA. Interfere with metabolic pathways. Again, and you'll see here in just a little bit, this is not necessarily the most common route that medications typically target. Why is this not necessarily a common target of antimicrobial medications? What have I told you, or what have you learned, or what do you remember about bacteria? Why don't we have a lot of medications that are interfering with metabolic pathways?
Their metabolic pathways are similar to eukaryotic cells.
Right. Perfect, right? There's a lot of similarities when it comes to metabolism between prokaryotic cells and our eukaryotic animal cells, or even other eukaryotic cells. And so, this kind of gets tricky if they do a lot of the same things we do, if you make that as a target. all right? And then interfere with cell membrane integrity, all right? And this is going to be really great for gram-negative bacteria, right? Because they have multiple membranes. And so there's different ways. And you can look at all of these and see, like, hopefully you can tell me why you think this might be good and why they might be bad based on what we've learned in previous units. So this is just another little diagram, but it puts a picture with it. And I do kind of like this because it lists some common classes of antibiotics under them. I'm not going to expect you to memorize, necessarily all of these antibiotics. We will talk about a couple different antibiotics from these different groups when we talk about what's commonly prescribed in the US. Those you will be responsible for, but I'm not going to say, okay, list the 5 targets of antibiotics and then every class of antibiotics is associated with them. No. Some of these, though, may stick out to you because you may have had them before, all right? So a lot of you have probably had some type of beta-lactam antibiotic. This is the penicillin family. And some of you actually more than likely have had some type of protein synthesis. So again, azithromycin, part of the Z-Pak, it actually falls under this category of microlids right here. And so you guys have more than likely probably had some of those before. But again, cell wall, right? Nucleic acid synthesis, cell membrane integrity, metabolic pathways, right? We'll talk about folate biosynthesis in just a second, and then protein synthesis, okay? So, I guess this is a little early because we get into this more next unit with kind of epidemiology and public health. But this is data from the CDC from 2016. And note too, I know this is a few years old, but I actually I think got this data last year. When you have massive data sets like this, when you collect them and when you publish them could be a couple of years apart, right? So this is actually very, recent data. And so you can see some of the different antibiotic classes that fall under those 5 categories that we just looked at. And so see penicillins, cell wall, microlids, protein synthesis, cephalosporins. Also, I believe are going to be cell wall, fluoroquines or fluoroquinolones are going to be targeting nucleic acid synthesis, right? And then beta-lactams, right? So these are going to be kind of penicillin-like, or at least go after cell walls like penicillin, right? So this is the class, all right? And then these are the specifics. So amoxicillin, right? Azithromycin, right? So this is that Z-Pak. And so you can see individual antibiotics. It's one of the most commonly prescribed. Amoxicillin with clavulanic acid. We'll talk about what that does here in just a second. And we're going to talk specifically about these 5 antibiotics in the next few slides, all right? But you can see, right, penicillins are still the most commonly prescribed antibiotic, right? So this is prescription rate per 1,000 persons. So the most commonly prescribed antibiotic. And then macrolids, right, which are going to interfere with protein synthesis, are the second most commonly prescribed. And then if you go down and look at individual antibiotics, not surprisingly, right, amoxicillin, which is in the penicillin family, is the most commonly prescribed antibiotic, followed by azithromycin, which would be in the microlids family. There are the most commonly prescribed. All right. So let's talk about some of these different antibiotics. Let's talk about amoxicillin. All right, so amoxicillin is a part of the penicillin family and is broad spectrum, right? It inhibits cell wall synthesis, right? So it is a beta-lactam antibiotic. We'll actually look at what that means in just a second. It's referring to a specific chemical structure of these different of beta-lactam antibiotics. If you go back and look at that last chart, right, you have the clavulanic acid, and this is commonly given with amoxicillin, and it is a beta-lactamase inhibitor. All right. So what do you think beta-lactamase does to a beta-lactam antibiotic? Laura, what do you think a beta-lactamase does to a beta-lactam antibiotic?
Breaks it down.
Breaks it down, right? Beta-lactamase, right? It's an enzyme that breaks down beta-lactam, right? Which is part of a lot of antibiotics. This is actually a mechanism that bacteria use, right, to resist the effects of medications. And so basically we're kind of trying to get one step ahead of the bacteria, knowing that some of them have beta-lactam ACE to break down our medication. So we give a beta-lactamase inhibitor along with our antibiotic that allows the antibiotic to do its job without being affected by the enzyme beta-lactam ACE, right? So it increases the effectiveness of the antibiotic because the bacteria is not able to break it down as efficiently, right? So amoxicillin is commonly used to treat pneumonia, bronchitis, infection, ear infections, nose, throat, urinary tract, skin, right? Again, this fact that the idea that it's broad spectrum, it is very commonly, prescribed to just treat a whole host of different issues. All right. All right. Here's cephalexin. It's part of the cephalosporin family. It is also broad spectrum. The thing that's nice about this antibiotic is it's also part of the beta-lactam family. But because of its chemical structure, it is actually naturally resistant to beta-lactamase, all right? And so you don't have to give, right, necessarily additional beta-lactamase inhibitors with this drug because it does it just based on its structure. We actually are on the 5th generation of these medications. And as we've gotten farther and farther away from the original cephalexin and cephalosporin kind of antibiotic, they've actually become more narrow spectrum, and these newer cephalosporin antibiotics are actually more effective against gram-negative, all right? But like penicillin, they're going to inhibit cell wall synthesis, right? And again, if you're a beta-lactam antibiotic, whether you're penicillin from the penicillin family or a different class of antibiotics, right, your kind of game is to interfere with cell wall synthesis. And here you see respiratory tract infections, urinary tract infections, bone, skin, ears, genitals, right? Again, the idea that they're broad spectrum means they're going to be used often for a lot of different things, right? And hopefully, you're picking up on the fact that of the specific antibiotics that are prescribed, a lot of them are broad spectrum. And so oftentimes when you go in and doctors say you have a bacterial infection, the majority of the time they're not actually culturing the bacteria, right? They're just kind of saying you clearly have some type of infection based on your white blood cell count and some other tall tale signs. So we're just going to give you this antibiotic. All right. Inhibition of cell wall synthesis. Again, beta-lactam antibiotics is what we've been talking about. This is the beta-lactam ring. So here's penicillin and here's cephalosporin. All right. Again, we know penicillin, right? It's great against gram-positive. Cephalosporin, I just told you, is broad spectrum, or, right, the newer generation of cephalosporin antibiotics are great at targeting gram-negative bacteria, right? So they both share this beta-lactam ring, but you can see that after that, the structure starts to differ a little bit. And you can see here 2 different side chains that are on cephalosporin. So the R, right, those of you that remember chemistry, this can be a lot of different things that we can add on to those medications. And this is one of the ways that we modify current antibiotics, right? Is we add different side chains onto them to try to get different activity for those drugs, right? When we talk about beta-lactam antibiotics, right, they are preventing the cross-linking of adjacent glycan chains. So if you remember in your cell wall, right, you have NAG-NAM, 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 right, altering molecules of NAG and NAM, and then you have sheets that are essentially laying on top of each other, right? And those sheets are going to be connected by the NAM to NAM linkages, all right? And so beta-lactam antibiotics prevent, right, the linkage of NAM to NAM, right? So they don't allow you to stack those sheets on top of one another. And what beta-lactam antibiotics are doing is they are actually competitive inhibitors of enzymes that are known as penicillin binding proteins, right? And penicillin binding proteins are basically enzymes that are facilitating the linkage of NAM molecules, right? So you interrupt, right, the enzymes that link NAM molecules on adjacent sheets of peptidoglycan together. All right, so that's what it's telling you right down here, right? Penicillin binding proteins catalyze the formation of peptide bridge between NAM molecules, right? So you're interfering with their ability to join NAMs on adjacent sheets of peptidoglycan. All right. You guys already pointed this out to me, right? Beta-lactam antibiotics. If a doctor is prescribing one to you, he is not going to give you a bacteriostatic antibiotic in addition to this, because you guys already told me, right? In order for a beta-lactam antibiotic to do its job, the bacteria needs to be dividing, right? So in this case, right, you know, these antibiotics are bactericidal. They are killing dividing bacteria, right? When it comes to drug activity, right, we've mentioned some of them are great at gram-positive, some of them are great at gram-negative. Then we have broad spectrum, which are good at both. And sometimes, right, you'd say, well, why can't we just prescribe, you know, why aren't there just 1 or 2 beta-lactam antibiotics? Well, what's really interesting is that those penicillin binding proteins that are joining the NAM molecules together on adjacent sheets of peptidoglycan, they differ amongst different bacteria, right? There's differences. And so, you know, we say penicillin binding protein, but penicillin doesn't work on all bacteria, right? And so not all antibiotics, or you need different antibiotics that target different penicillin binding proteins, right? Because if you're not binding to this penicillin binding protein, you're not preventing those NAM linkages from happening, and you're not able to kill the cell, all right? So that's why we have a lot of times different medications. Different medications interact with different penicillin binding proteins, allowing them to be effective and do their job and remove bacteria, all right? Now, we already mentioned this, right? But some bacteria synthesize beta-lactam ACE, right? And so this is a natural mechanism that some have in order to combat some of these beta-lactam antibiotics. This is information that could be shared potentially during horizontal gene transfer, all right? Maybe bacteria A, right, is susceptible to, we'll say, methicillin, all right? Bacteria B is resistant to methicillin. They get together in one of the horizontal gene transfer fashions, and that information is shared. And now bacteria A that was susceptible is resistant. All right, so this is one of the genes, right, for a beta-lactam ACE that could be shared between bacteria that would allow something to be resistant that wasn't before. All right, when it comes to beta-lactam ACE, again, not surprisingly, there's different penicillin binding proteins. Different bacteria have different beta-lactam ACE also. So you have different proteins that are helping to build the cell wall, right? You have different proteins, enzymes that are helping to combat the medications that come in, right? So penicillin ACE, right, is a specific beta-lactam ACE that's present only in the penicillin family. You also have what we call extended spectrum beta-lactam ACE. And so again, there's a wide variety of beta-lactam ACEs that exist amongst bacteria. And so, you know, you can't necessarily just give a universal beta-lactam ACE inhibitor and hope that you hit the target. Sometimes it'd be really specific. And what kind of complicates the matter when it comes to beta-lactam antibiotics is that we see a wider variety of beta-lactam ACE in gram-negative bacteria. And so gram-negative bacteria have a couple of things working in their favor when it comes to beta-lactam antibiotics. You know, they have that outer membrane that provides protection, right, from the drug interacting with their cell wall. And then it turns out that even if that medication gets through, they have a larger or wider variety of beta-lactam ACE. So even if you were able to reach the cell wall of gram-negative bacteria, they're actually probably going to be better at clearing that antibiotic, all right? This is just showing you, right, when I talk about the penicillin family and I talk about different side chains. So if you remember in that first picture that I showed you, this is where you had that R right here. And this is the basic penicillin structure. So here's your beta-lactam ring, right? And if you go down here and you look, depending on what you change that R, that side chain out with, you get these different antibiotics, right? And then you see, right, that the activity or the resistance or what they're resistant to certain drugs, you can see it change just by changing the side chain, right? So this is kind of what we're interested in a research setting. In some cases, is can we alter the side chain to give these drugs different activities and maybe overcome some of the resistances that bacteria have or express. So we talk about beta-lactam antibiotics. We don't really, I'm not going to give too much time to glycopeptide antibiotics. They are also interfering with the joining of adjacent NAM molecules like beta-lactam antibiotics, but they are just doing a little different, right? So here you prevent the enzyme that's joining the NAM molecules together. Here, right, for glycopeptide antibiotics, you actually essentially are binding to NAM, and even if the enzyme's there to link them, it doesn't have access to that NAM molecule, so it can't get to its target to join them together. All right, and then another cool one is bacitracin, and this antibiotic actually prevents the bacteria from sending out NAG and NAM to outside of the cell membrane to be assembled. So remember, bacteria, right? A lot of activity actually takes place outside of their cytoplasmic membrane, and so they have to pump, right, or move a bunch of things across their cytoplasmic membrane to get to their cell wall area. And so here, what you basically do is you prevent the building material from reaching the construction site, right? So that's what's going on right here. So kind of a cool way to go about it. Mm-hmm. So let's talk about azithromycin. So this is, part of the Z-Pak, right? These are inhibiting prokaryotic protein synthesis, right? 70S ribosomes. And so the idea here is that whether you have a cell wall or not, azithromycin is going to be able to affect those bacteria. That's why it's a great kind of broad spectrum. So the doctor hasn't, identified the exact bacteria that's causing the problem. This kind of covers their basis. All right. Azithromycin also is really good. If any of you have had a Z-Pak, it stays in your system longer, and so you don't have to have as much individual doses of the drug. Specifically of the 70S ribosome, right? It's targeting the 50S subunit. Remember, it falls under that group of macrolids. And what azithromycin and other macrolids are going to do is they're going to interrupt translation. All right. So basically, the bacteria cell has made mRNA, and it's getting ready to translate that mRNA into proteins, right? That are necessary for cell division, necessary for the activity of that bacteria. And this stops that from happening. All right. And so here again, pneumonia, bronchitis, STIs, right, formerly known as STDs in my time, I guess sexually transmitted infections is not as harsh as sexually transmitted disease, right? And just pretty much any type of infection, again, because they are targeting something that almost all bacteria have. HIV patients, right? This is commonly prescribed to them. We're worried about a lot of infections in HIV patients, but mycobacterium and mycoplasma infections are something that we worry about to a much higher degree in HIV patients. And if you're allergic to penicillins, this is an antibiotic that's commonly prescribed, right? Because typically patients won't have an allergy to it. And so when you are inhibiting protein synthesis, right? If we're talking about something like azithromycin, right? Or the microlids class, these can be bactericidal or bacteriostatic, right? And so, the idea would be, if we're talking about it being bacteriostatic, if you go back to your growth curve, essentially what it will do for some bacteria is it will kind of suspend them in that stationary phase, right? That was at the top of the growth curve when they would use nutrients. Kind of the same basic principle here. They're no longer able to synthesize protein, which means they're no longer able to divide, all right? And so it's almost like you're inducing, right, a stationary phase, and then which then allows them to be a target medication, right? But you can also imagine in some cases how inhibiting protein synthesis could be deadly for the bacteria. So within, microlids and other antibiotics that inhibit protein synthesis, you can see some that are both bactericidal and bacteriostatic. And again, you guys already know this. We talk about this M&S ribosome, right? You guys have told me multiple times before. This is what we have in our mitochondria. And so even azithromycin and other macrolids and other enzymes or other medications that target protein synthesis can eventually be toxic, right? When we talk about protein synthesis, we talk about azithromycin targeting translation, right? You can prevent the initiation of translation. You can prevent the continuation of translation. Tetracycline, which we talked about in seal finger disease, right? It actually inhibits transfer RNA, right, which is bringing amino acids to ribosomes as they are building proteins, right? So you have a lot of different possibilities that exist there. And, this is looking at all the, again, I'm not going to ask you to memorize these. I just do want you to know when we talk about inhibiting protein synthesis, there's different ways that you can do that. And the different drugs, for that. So we just talked about azithromycin, which falls into this category. But here you can see some other ones. So tetracycline, right? That's from the seal finger disease, right? And again, I'm not going to ask you to regurgitate this slide, but I do want you to know some basic ways that we inhibit protein synthesis and what we're targeting in prokaryotic cells. Okay. So this is it for metabolic pathway interference. There are two drugs in this category, sulfamethoxyl and trimethoprim. Sorry for my pronunciation of those drugs. These are given in combination typically because they are targeting the same pathway. You can see here just at different points. All right, so sulfa drugs target this pathway early on. Trimethoprim targets it later, and this is kind of nice to give in combination because if somehow bacteria is able to develop resistance to the sulfa drugs, well, then you're going to be able to target farther down the chain. This is actually interfering specifically with the folic acid pathway in bacteria. Unfortunately, we also make or folic acid is important to us, and so again, there's not a lot of metabolic pathways that we target, but when you target these pathways, you can see here that you're interfering in a lot of ways with their ability to synthesize and make new nucleotides that will be used later down the road for replication, right? And in this case, right, these are some of the few drugs, because although we have similar pathways, right, these actually don't interfere with eukaryotic enzymes, right? So these are great for gram-positive and gram-negative urinary tract infections, middle ear infections, pneumonia for HIV patients. But again, one of the few drugs that's targeting metabolic pathways because it's hard to find targets, just like you guys said a little bit earlier, due to the fact that prokaryotic and eukaryotic cells share so many similar metabolic pathways. All right, so let's talk about inhibition of nucleic acid synthesis. These are a few drugs or a few classes of drugs that do this. Fluoroquinones, right? They inhibit topoisomerases, which prevents replication and transcription. What's topoisomerases doing? Does anyone remember? I can even take a stab in the dark. Would anyone look at?
I think they help unwind DNA that are twisted or something like that. I'm not so
Perfect.
sure.
Yes. Thanks, Toomey. Right. Toomey's right. They help unwind DNA. And so if DNA is not unwound, right, then DNA polymerase for replication can't bind, and RNA polymerase for transcription can't bind, right? So by preventing the unwinding of DNA, you prevent the bacteria from making copies of genes or other things, right, that it needs. Or, well, genes are what it needs, but you get what I mean right there, okay? So ciprofloxacin, right, is often used here. So gonorrhea, right, which is an STI, typhoid fever, which I believe a couple of you are doing typhoid, infectious diarrhea, some of these sound unpleasant, right, prostate infection, the plague, right, hopefully you don't encounter that, or anthrax, right? So these are pretty important antibiotics. Rifamycins target RNA polymerase specifically. So rifampin is a drug that's in this category. So tuberculosis, leprosy, right? So targeting those mycoplasma infections, and this makes sense, right? Because these bacteria right here don't have cell walls, and so you need something that's targeting a different aspect of them. So nucleic acid synthesis is a great target. Metronidazole. So if any of you have, these are really common in inflammatory bowel disease. So if you know someone with Crohn's or colitis, this is a great medication for them. You also see C. diff on here. So this is commonly prescribed for patients with C. diff. This medication is great at targeting anaerobic organisms, a lot of times that live in your gut. All right, so if you have infections in your gut, your small intestine and large intestine, this can be commonly prescribed. Also, bacterial vaginosis for women, this might be prescribed. Interestingly enough, too, this drug, when you take it, you can't drink alcohol, right? So drinking alcohol while you're on metronidazole causes projectile vomiting. And so it can also be prescribed by doctors that are trying to help patients give up alcohol, right? Because projectile vomiting, at least for some people, can be enough of a deterrent. And interestingly, the common name for this drug is Flagyl. So at least it has a fun name, right? All right, we also go to interruption of cell membrane integrity. Again, please don't overthink this. I'm not looking for a deep philosophical answer. Why do we also, along with metabolism, have very few drugs that are targeting the cell membrane of prokaryotic cells?
Because we have cell membranes.
Because we have cell membranes, right? And there are differences between a lot of times maybe some of the proteins, the transmembrane proteins that are present in the cell membranes of prokaryotic cells and eukaryotic cells. But for the most part, the basic structure of our cell membranes is very similar, all right? And so this is not necessarily a common drug target. You have a couple big groups, daptomycin, right, which is great for blood and skin infections. And you also have polymyxin. So if you've heard of polymyxin B, that's an antibiotic that's, if you've ever had eye drops, like, so if you've had pink eye and they think it's bacterial, I could almost guarantee you that there's polymyxin B in those eye drops. All right. So again, not very common, but we do have some of them. So let's talk about antimicrobial resistance. This is actually a link to another TED Talk on antimicrobial resistance. It's pretty good. So if you have time, I'd go and watch it. But that actually, this was kind of mentioned a little bit in the video, and I've mentioned this before, but what this slide is doing is it's addressing the idea of what happens when, well, I'll look at, I can see Noelle on my screen right now, and I know that, you know, they have strep throat every other week in her house, so that they probably have antibiotics, you know, that are present in their house on a regular basis, right? So what happens, right, when you don't finish, you know, all of your drugs? And I know this came up a little bit earlier in a conversation. So here, right, you see, what do we have here? 7 bacteria, right, just as an example, and they're saying that most of them are S, right, and that S stands for susceptible or sensitive, and the R is resistant. So you see in this population of bacteria, the majority of them are susceptible, right, or sensitive to whatever antibiotic has been prescribed to you, all right? And so when you start taking that antibiotic, typically most of us would probably say that within 24 hours, often, or maybe even less, right? If you would take it before bed and then you wake up the next morning, often you're feeling better. And the reason you're feeling better almost immediately is because that medication works very quickly on these susceptible or sensitive bacteria. All right. And so we've talked about it, and I know you guys are all intelligent, right? But maybe you have, or maybe someone you know has asked, well, if I'm prescribed antibiotics for 10 days and I start feeling better after 1 or 2 days, why do I need to keep on taking the drug, right? And the reason you need to keep on taking the drug is because of this resistant bacteria here, right? In most cases, right, and again, there's always exceptions to the rule. So the article that I showed you today about the woman that died a couple of years ago in Nevada, she had really resistant bacteria, right, to every antibiotic available in the United States. But in the most cases, resistance is not invincibility. It just means, that they require a higher dose or more persistent dose of that medication. And so, if one of Noel's brothers, right, with his strep throat, decides to not finish his medication, he's by day 2, day 3 of 7 days, he feels great. He's like, I'm through with this, right? He has likely, or there's a chance he's failed to kill, all right, the resistant bacteria. And when you do that, right, although you feel better, in a few days, right, that resistant bacteria now divides. You start to feel ill again. And, oh, by the way, now the majority of the bacteria in that population are resistant. So they're either going to require a different medication or an even longer prescription of the same medication you had before, all right? So one of the things I want you to be thinking about in this unit, and this becomes a public health issue, which again feeds nicely into our next unit, is, what are things that you can do as a patient? And here's one of them that's relatively simple. Finish your medication to prevent antibiotic resistance, or at least slow it down. And those of you that go on to maybe be physicians or PAs or nurse practitioners, right? What is your level of responsibility in this? Okay. I really like my doctor, and he definitely wants to make you feel better, but he also, as long as he feels like you are not on death's door, he will take a little bit of a, hey, let's wait and see. All right. My doctor will prescribe an antibiotic, but then he'll say, why don't you, right, wait for 1 or 2 days and see how you feel, right? And if you feel better, then don't go and get the antibiotic. If you don't feel better, then go and get the antibiotic and call me and let me know, right? So responsible practice goes both ways. All right. So let's talk about resistance. What's happening on a cellular level, right, that allows a bacteria to be resistant? And here, right, is a non-resistant cell. So this is what we hope most of the cells are that you guys, are using your antibiotics for. And so this is really generic, right? It just says you have an antibiotic. It has a target. Remember, that target could be a lot of different things in that bacterial cell, right? This is just kind of a generalization. The antibiotic binds to the target. Once the antibiotic is bound to the target, it then either inhibits cell growth so the immune system can take over, or it directly kills the bacteria and aids the immune system that way. But either way, right, the antibiotic finds its target and you feel better. What we see in resistant cells are basically 4 different kind of mechanisms that are leading to direct resistance of drugs, right? So one, increased elimination, right? You have transmembrane, not you, your bacteria or pathogenic bacteria, some have transmembrane proteins that are great at pumping out medication, right? So if you think about this, right, if I we'll put Allie, she hasn't had anything bad happen to her in microbiology class in a couple days, right? We put her in a boat in the water, right? It's a small boat, and I give her a little bucket, and I poke a small hole in the boat, and it starts to fill up with water. What does Allie do? Allie, or what would you do?
I guess shovel the water out.
I guess that's what I would do. That'd be a good idea, right? There's a lot of other things that might happen, but if any of you might have ever been in a boat for whatever reason that has a small hole in it, right? You shovel the water out, and that's what these bacteria do, right? The medication comes in, and they shovel it on out, right? And if you're shoveling it out, it has trouble hitting its target, right? Antibiotic inactivating enzyme, right? So this is we talked about beta-lactamase. So beta-lactamase, right, binds to the antibiotic, inactivates it, and it can't hit its target. All right, we're kind of going clockwise here. Alteration in target molecule. So if an antibiotic is looking for a certain target, some bacteria get really smart and they slightly change the structure of the slightly change the structure of the target. And so the antibiotic no longer can bind to the target, right? And then decreased uptake, right? So remember we talked about poron proteins not just being these big channels that allow things in and out. They do have the ability to be selective. And so in this case, poron proteins can close in the presence of antibiotics and prevent them from even entering the cell. And then that antibiotic is not able to get to its target. Do we see this decreased uptake? Is this in gram-positive or gram-negative bacteria, and why? Anna, can you help me out?
Is it because of the different thickness of the membranes between the two?
That's close, right? You're in the right ballpark. Someone help her finish that off to get a 5 out of 5.
Poron proteins are found in the outer membrane of gram-negative bacteria.
Perfect, right? Poron proteins are found in the outer membrane of gram-negative bacteria, all right? Because you have that outer membrane that you have to get things across in order to interact with the cytoplasmic membrane, all right? So this is only something in this case that we'll see in gram-negative, because gram-positive, right, have that thick cell wall. They don't have poron proteins, right? Perfect, all right? So the ability to do these different things, right, is because of different genes, right, that lead to the formation of different proteins and different enzymes. And so the ability to do these things is what's shared in horizontal gene transfer. So again, I'm looking at my screen and I see Elena and I see Rachel S., right? And so let's say Elena has a cool little trick that she's learned about how to neutralize an antibiotic, all right? She's a very sharing and caring person. So she shares that information with Rachel, who before had been susceptible. She had been Rachel S., and now she's Rachel R., right? Rachel resistant, because of that sharing of information. So this is the information that's shared. When we talk about what are the genes, or what do the genes allow you to do that are being shared, these are the different things that they can do. Okay? Now, he mentioned, right, kind of another way, right, or I guess specifically how resistance is happening. And so this top chart, right, that I like, it shows you sometimes, right, that resistance is just spontaneous, right? There's a spontaneous mutation within a gene, right, of a prokaryotic cell that allows them to be resistant. And then subsequent, right, division, right, via binary fission of that cell that has that beneficial mutation, all right, allows, right, that mutation to keep on going and survive, right? Again, not all spontaneous mutations are bad. Not all spontaneous mutations lead to good things. And some case of bacteria has a spontaneous mutation that's not beneficial, right? there's a good chance that cell could die. So in this case, the bacteria got lucky. It finds a spontaneous mutation, or it has a spontaneous mutation that ends up being helpful. All right. You also have, right, we've talked about horizontal gene transfer that can occur in a number of ways, right? Often, in this case, if there's resistance shared, it's via an R plasmid, right, a resistant plasmid. But please note, right, that even with this spontaneous mutation, let's say for whatever reason this cell dies, right, and it's lysed, well, then we could have transformation that could spread that information. Or we could have transduction, right, a bacteriophage infects. And so all of these, right, even if it started off as a spontaneous mutation, if it's beneficial, that information could potentially be passed on to other bacteria in the future. Okay, this is just, again, kind of another little public service announcement, but this is data gathered by the CDC. And you know, what they estimate is that about 70% of antibiotics prescribed are actually necessary. 30% are unnecessary prescriptions. And they're even telling you within those 70%, we probably could do a better job at prescribing more specific medications and even adjusting better the dose or the time that we prescribe those medications, right? And we'll find out in a little bit. Again, it'll probably be a little while till this data is available, but their goal, you know, by this year was to reduce inappropriate outpatient antibiotic use by 50%. They may actually achieve that, I guess, because we don't have as many people going to the hospitals right now or doing nearly as many surgeries or operations. And so I would say that's an artificial accomplishment of the goal. You know, that's the fun thing about statistics, and I guess even pandemics, is that, you know, you could theoretically report that data and say that you've done a good job. But I don't know what those numbers will look like. Or if they report that data, hopefully they'll report it with a little asterisk and say, but one of the reasons that we reached this number is because no one was allowed to go to hospitals anymore during the pandemic. Right, so who knows? We may be artificially getting there this year. I also just wanted to show you, this is interesting. And again, this data is relatively actually recent. You can see this was published in the 2016 article. Again, it takes time sometimes to collect these massive data sets. But this is antibiotic prescription per 1,000 in the year 2015 in the United States. And so you can see the darker reddish hue means that we're prescribing more antibiotics. The lighter hue, right, means less antibiotics. And what's interesting, when you take a bunch of these heat maps, if you guys were to go on the CDC's website and look up obesity heat map, heart disease heat map, a lot of the areas that are the darkest here, right, a lot of these patterns would be similar. So it kind of sometimes gives you perspective of the overall health of certain populations. And what you can see here, if you're from this area or anywhere in this area, is that if you're from the Southeast, you more than likely are leading in almost every major category that's measured by epidemiologists, right? So here we have Tennessee, and you can see prescriptions per 1,000, right? So basically, you know, what they're saying is if you would look at our population, not necessarily an individual basis, but the number of antibiotics that are prescribed in Tennessee means, based on these numbers, that each person had at least one antibiotic prescribed to them, which we know is not true, right, in a lot of cases. And so some people may have a lot more. But if you average out all of the prescriptions that were given in Tennessee, there's at least one per person. And so, you know, and this is not, you know, one of the things we have to be careful too is we don't want to antibiotic shame either. I mean, sometimes, you know, these are necessary, but there certainly are things that we can do to help with this. And then this is just, you know, to give you perspective. And I think some of this is not overly shocking to you, right? It's basically saying, by profession, who's prescribing the most antibiotics, right? And so, you know, for me, I would actually just link these two together, primary care physicians and PAs and nurse practitioners, right? To me, these are all the same. And I think in the future, we probably will just group those together, but not shockingly, right? The doctor that you go to, or the PA or the nurse practitioner that you go and see as your primary care physician, they're the ones that are prescribing the most antibiotics. Surgery, right? Again, not uncommon after you've had surgery, especially depending on the invasiveness of the surgery, you are prescribed antibiotics. Again, though, this is actually a pretty big target area because it was kind of common after surgery to just load, and still is a lot of cases, I guess, to load the patient down with antibiotics, which again, you don't want someone that's just had surgery to get an infection. But now this is even being kind of looked at, right? Is there a lower dose that we can, prescribe for a shorter, right? There's a lot of things to look at. This is one that I actually found most interesting. The top 4, dentistry, rounds out. And again, I can't speak from experience. I've never had an antibiotic prescribed by a dentist. I didn't know this was something that they commonly do. And then you can see some other ones down here, ER visits, right? Dermatologists, all right? And then you have, I'm not exactly sure what other would be, who else has the ability or the power to prescribe antibiotics besides, groups here, but maybe this is getting really specific and saying like ENTs and, doctors that have a specific area of interest, or podiatrists or proctologists. I don't know, right? But it just kind of puts in perspective, right? Where are our antibiotics being prescribed at higher levels? And one of the things that I want you to use this information that we talked about today, and in the video that you watched and the video that you should watch that's on our Moodle page is, what can we do to slow down antibiotic resistance? And then in that video too, what I want you guys to pay attention to are what are alternatives to antibiotics, or what can we do to make our current antibiotics more effective? And in the video that I have posted on Moodle, he kind of presents some interesting cases and interesting situations for you guys to think about. All right. I think that's it. Awesome. Tomorrow we'll start on epidemiology, and I'll try to incorporate, depending on how much reading I can get in tonight, some things on COVID-19 into that lecture. And we'll also use HIV as kind of a great example of the importance and the effectiveness of epidemiology. All right. So with that being said, it's 10:04 according to my thing. Let's come back together at 10:15. I want to talk about your project briefly, and then we will go from there. All right.